Hello, thank you for coming. My name is Paula Trzepacz, and I'm here on behalf of Todos to talk about LymPro, the based blood test for Alzheimer's disease. I've been both a clinician and a researcher in Alzheimer's disease at academic center. This is just the things I say today will not be can't be promises about the future. So we know that the Alzheimer's brain has certain classic neuropathologies such as amyloid and tau abnormal protein buildups, but also there's shrinkage and atrophy of the brain from a cell loss, as you can see up there. And in particular, I don't know if I can point. Oh, in particular, the hippocampus, which is normal here, is very shrunk in Alzheimer's disease. So the LymPro test is a simple blood test. And it reflects the neuropathology found in Alzheimer's disease. So the question is, if you could get the test, would you want to know? Why do we need a blood test for Alzheimer's disease? Well, we need to increase the accuracy of diagnosis, which in the hands of experts is at best about 70% if you're in the dementia stage and 50% at MCI. So that's like flipping a coin at MCI. It would be a much more convenient way to get biological information, and if it was a valid surrogate or a proxy for some important neuropathology in the brain, then you could get information before waiting for autopsy. And hopefully, since it's a blood test, it can be cost effective, much more widely available, including around the world. It could help also to identify patients more specifically for Alzheimer's research. So at Lilly, I looked back at some of our C-mediated and solanezumab data where we did have some amyloid data on them because it wasn't required at the time we were doing those trials. And we found out that a whole bunch of people in those trials didn't have amyloid. So they could have a frontal temporal dementia or something else, and then you start to wonder, well, why didn't their drugs work? So there's a recent study that came out in JAMA, which you guys may have read about. The IDEA study is an imaging study for amyloid, and there are 13,000 patients in the study, and they reported on over 11,000 who had data. And what they did is they went to specialty dementia clinics throughout the United States. They had symptoms for either MCI or dementia, and they were evaluated the usual way by these experts clinically with their various tests and given a diagnosis, what was wrong with them and what caused. And then they got an amyloid PET scan, and then depending on the results of that, the diagnosis would change. And as it turns out, 25% of the patients who were diagnosed as having Alzheimer's did not have Alzheimer's after the scan. They had a negative scan. And 10% of those who were diagnosed as not having Alzheimer's did have Alzheimer's, so they had a positive scan. If you look down here, this would be an example of a positive scan in Alzheimer's with a lot of red and yellow for the amyloid plaque, which is these little blobs of proteins in the brain. And the negative scans would have looked like this, so they wouldn't have the evidence of the plaque. One of the confounds is these patients who were clinically normal, when you have scans, not necessarily in this study, but clinically normal, but they had positive amyloid. And so it's a confound if you want to know, do I have it or don't I have the pathology? And you're normal. These people could go for 20 years, or they may never get dementia. So this is a real issue with this type of a scan, looking for amyloid. There are other currently available biomarkers that are used in clinical practice, and these include the amyloid plaque. This is just a different version where it's more black, is more amyloid. We also have regular PET with FDG, which is radioactive glucose, and it shows what the metabolic activity neurons, which ones are active and busy. And when you find a classic pattern in the posterior region, including the hippocampus and Alzheimer's, where you're not getting this metabolic activity as you should here. It's not entirely as specific, though, as looking at an amyloid scan. Then you have MRI neuroimaging where you would show, as compared to a normal, you see atrophy in the brain substance, and especially here in the hippocampus, just like I showed you on the pathology in the first slide. The alternate to imaging would be cerebrospinal fluid, which is a lumbar puncture in your lower back. They take out fluids. If you've got low amounts of soluble amyloid beta and high amounts of tau you end up in this box. And compared to autopsy, with these solid circles, well, that's pretty good. The problem, and over here is pretty good where your normals in the open circles are not in the abnormal range, but the problem is the confounds in there, and also that the standards are not well standardized across labs, so it's not as well standardized as the neuroimaging. But that's what you have today that's available. So we need an alternative, particularly so we can distinguish, do I have Alzheimer's or not? So what's an alternative mechanism that relates to the atrophy and the neuronal deaths that we see as one of the characteristics of the disease? And here you'll see that most body cells make copies of themselves to replace old damaged cells. So they grow fatter, and then they're in the nucleus, the chromosomes and DNA duplicate and divide, and then you end up with 2 new cells. It happens in your skin, it happens in your hair follicles, it happens in your gut, it happens in your hepatic system. And for me, unfortunately, it happens right here for adipose, and I just wish they wouldn't keep doing that, but they do. But the contrast is in mature brain cells, neurons, they're not supposed to divide and do that, okay? So, looking at a similar picture up here, which shows the same division of the cell and growth, except that it's showing the chromosomes, so that the DNA, the chromosomes are duplicating, and then when they divide, each cell, each daughter cell has the right amount of the DNA and chromosomes. And the way this gets started is there's some kind of protein, antigen or mitogen, that says, go, it stimulates and activates this process. And that whole cascade of events occurs, like building a car in a factory or something. But if something goes wrong along the way, they get a stop command, because there are different kinds of repair proteins or halt proteins or stoppages to keep that from happening, because it's going awry and you don't want to keep manufacturing a bad cell. So, as I said, neurons in normal brains do not enter this cell cycle. They're mature, differentiated, they don't do this, or at least they're not supposed to do this. And they don't even have all the proteins anymore from embryogenesis. They had all this stuff and they don't need it anymore, so they don't have it. So what happens is, in Alzheimer's brains, we know that there's abnormal cell cycling. They re-enter the cell cycle, these neurons, and they're not supposed to. And but because you don't have all the machinery, you end up with too many copies of DNA. You end up not repairing things, and you end up dying. So they shouldn't do it. They're supposed to be resting, and they don't. So you end up with multiple copies of DNA. And here in the graph, you'll see there is a little bit of this multiple copies of DNA that you can see in immunohistology and brain slices. But AD at different stages has far more percent of this, far more frequency, which is not normal. So therefore, this brings us to the LymPro test, something that's unique and going to potentially measure this kind of neuropathology of the aberrant cell cycle re-entry in the brain of neurons. And as I mentioned, it's a simple venipuncture, just like any old blood test. Gets shipped out to anywhere with a central lab, and then they spin down for lymphocytes that then goes through a machine called the flow cytometer. So what it's going to measure is it measures the amount of a protein called CD69. It's on the cell surface of all lymphocytes, and it is a marker when the lymphocyte enters the cell cycle, this goes up. The density increases, and that can be measured, and it's a sign, it's a marker that someone has gone to the go sign, okay? And so that's what we're going to measure, what we measure in this test. So what happens is here's the flow cytometer, and you can see what it does is it actually lines up these white cells, these lymphocytes. They queue up one by one, and so very well behaved. They all queue up one by one, and then the laser shines through, and there's fluorescence of the proteins, and then the various proteins can be determined how much is in there, okay? So we're using certain lymphocytes, B and T lymphocytes, that have these little markers, little proteins on the outside. So there's CD3, CD4, and CD19. These are the specific ones that go through and get measured, and we measure the amount of this other receptor, CD69, that's the marker of entering the cell cycle. So we can measure all of that in this fancy machine. So what do we find? Well, the cell cytometry things look sort of like these. You get these blobs. And what we see here is that the normal subject's lymphocytes express a lot of the CD69. They've gone into the proliferation they're supposed to. They need to fight infections, right? That's what you want them to do. So that they're doing it when they're stimulated by a mitogen. They do that. But down here, you don't see that in Alzheimer's disease. Something's wrong. Something's off. And what this does is it reflects what's going on in the brain in terms of the cell cycle. They have lots of evidence of cell cycle abnormalities in entering the cycle. And what we find here is that you can see graphically that healthy controls are producing, in these two kinds of lymphocytes, more of the CD69. So this is a mirror in your blood of what we think is going on in the pathology in the brain. So, in summary, the LymPro test holds promise as an adjunctive test to diagnose MCI and dementia due to Alzheimer's disease. It's a routine blood test. It's convenient, a simple blood draw. Unlike PET scans, there's no radioactivity involved. No need for cyclotron or being basicity, and likely lower cost because PETs are so expensive. And so far, it looks like normals are distinguished from Alzheimer's on this test. And it's a seamless integration that could be, when we get more data, developed so that clinicians in the office could use this, the experts, and who knows, perhaps even help the family practitioners who have a terrible rate of accuracy of diagnosis. So our further studies include assessing relationship to amyloid PET, and we have some data on that. They're not ready yet to be presented. It also needs to be assessed as a staging tool, so it looks like may be a quantitative difference between MCI and Alzheimer's, but that needs to be looked at. And we found that it does distinguish Alzheimer's dementia from Parkinson's dementia, but we need to look at other types of dementias as well and see whether this could distinguish them. And if so, it could be very promising. So thank you very much for listening.